Hello, this is Lisa Poole. I'm recording the video for the Unit 2 case connection for the biochemistry course at UNE. I've selected Case 2 and Case 4 to review in this video. Case 2 involves the regulation of glucose uptake by the cell. It involves cancer cells versus regular cells, specifically this tumor in the brain. In normal tissue, glucose uptake is regulated by capital GLUT, GLUT. In mammalian cells, GLUT1 facilitates transport of glucose across plasma membranes. GLUT1 is a uniport protein that are encoded by SLC2A1 genes and transport glucose into the cell down the concentration gradient. It has 492 amino acid residues. It is ubiquitous. This is found in nearly all fetal tissue and also in many adult tissue. In many organs, it is found in the endothelial cells of the blood tissue barriers. GLUT1 shuttles glucose between blood and organs that have limited access to small solutes through passive diffusion. This will help maintain equilibrium. It won't increase glucose in an already high concentration. After birth, GLUT1 decreases in other isoforms like GLUT2, which regulates glucose uptake in the liver, and GLUT4, which regulates glucose uptake in the muscles and in adipose tissue. The messenger RNA, GLUT3, is found mostly in the brain, the kidney, and the placenta, although truly it's found in all tissue. It has a high affinity for glucose and ensures efficient uptake due to the highest calculated GLUT turnover. GLUT3 is also considered the primary neuronal glucose transporter. Otto Warburg observed in 1924 that cancer cells and normal cells metabolize glucose differently. Mutations in cancer cells result in glucose uptake that exceeds the bioenergetic demands of normal cells. Normal cells in an aerobic state have lower glycolysis rate. Cancer cells have the higher glycolysis rate, roughly 10 times higher than a normal cell. Thus, with higher energy, even in an aerobic state, a higher energy demand, even in an aerobic state, and they end up producing lactic acid that ferments in the mitochondria. This lactic acid cycle, cancer cells thrive. They thrive in acidic environments as well, and this perpetuates this issue. Energy is produced through conversion of pyruvate to lactate. The cell is signaled to switch to the pentose phosphate pathway, PPP. It is a parallel pathway to glycolysis, whereby excess glucose is diverted and utilized by cancer cells. It biosynthesizes lipids, nucleotides, and helps tumor cells NADPH, which helps tumor cells meet their massive metabolic demands. Case 4 will also be reviewed. This child has glycogen storage disease, specifically type 1 or von Gerke's disease. This is characterized by a missing or malfunctioning enzyme normally utilized to break glucose into glycogen in the liver. This disease changes how glycogen is stored and utilized in the body. The clinical exam on the infant reveals enlarged liver, inability to maintain a blood glucose in the normal range past feeding, a high lactate level. All are symptoms of this disease. Additional symptoms of the disease would include restricted growth, heat intolerance, excessive bruising, weak muscles, and high cholesterol in babies. Treatment includes frequent eating throughout the day to avoid low blood sugar, avoiding fruit and milk sugars which are not able to be digested, and sometimes using medicine to lower uric acid in the blood and the urine. Symptoms for hereditary fructose intolerance include diarrhea, bloating, gas, vomiting, abdominal pain, fatigue, and malabsorption of nutrients like iron. None of these symptoms this infant was displaying. Galactosemia signs and symptoms include convulsions, irritability, lethargy, poor weight gain, jaundice, and vomiting, none of which the mother was describing in this baby. Glucose 6-phosphate dehydrogenase deficiency is a type of anemia that shows itself with yellowing of the skin and eyes, fatigue, shortness of breath, enlarged spleen, dark urine, and rapid heart rate, none of which this infant was displaying. This infant is suffering from glycogen storage disease.